Welcome back, guys. It's Shahfahadkhan on the screen. Okay, so now we're back with the foundation module guidelines of first year MBBS, and now we are gonna talk about biochemistry. Okay, in fact, one of my favorite one of my favorite subjects, and if you had chemistry in O levels, well, or A levels, this is somewhat similar to chemistry, but is not actually chemistry. This is more like of chemical reactions going inside of our bodies. Okay, and you'll enjoy it because it does have mechanisms and the different enzymes and catalysts, etc. Well, the catalysts are actually the enzymes working in the human body that you'll be talking about, all the different translation, transcription, and all those basic biochemistry that we had in our MCAT, in our A levels, in our O levels, in our inter. We'll be doing and talking about all of that over here. So now let's get started without wasting any more time. Okay, so what is biochemistry? Okay, so biochemistry, what does it explain? It explains the biochemical. Bio means life, right? Meaning the chemical reactions occurring inside living bodies or living systems. So biochemical processes, the chemical processes occurring within living systems or living bodies are known as biochemical processes. So it actually explains all those processes occurring within living things, mainly concerned with at the molecular level. Okay, at the molecular level, at the with the molecular point of view or molecular mechanisms which are involved over there. Remember, in chemistry we had mechanisms for different chemical reactions that were occurring. Over here as well, the chemical reactions occurring in the human body, their mechanism, their molecular mechanisms are mainly described in biochemistry of all the different chemical reactions occurring in the body. Well, the section of body that we are concerned with, basically the biochemical processes of that section only in foundation. Okay, it's not in a lot of detail, but it is still a big chunk. Okay, it's still a big chunk, meaning all the topics are covered. There's just an overview of all the topics, but still it's a chunk, a huge chunk of your foundation module. Okay, then biochem of foundation shall cover almost everything covered in physiology and pathology and embryology. Okay, physio, patho, and embryo, all of them are actually covered in biochemistry, and actually they'll be reviewed in biochemistry again. So again, the stuff that you've done over there, it will become solidified once you reach your biochemistry and you start doing it. And in fact, biochemistry would become a lot more easier if you have already done your physiology and pathology and embryology before it, because of course you will already be familiar with the stuff. Okay, but again, the extra molecular details would come in. Yeah, and I do have the first year playlist on biochem, and I have given the description down, the link in the description, or maybe up there or here in the cards you might see it. But anyways, this is how it works. Okay, and a playlist has been given on a few topics which are in fact very, very important, and they're not new. You've already done them in your previous classes in inter or A levels. But again, the thing is that you'll still need to do the extra stuff that will be in here. So there will be the same stuff that you've done previously plus some new stuff of MBBS. But trust me, guys, you'll enjoy biochemistry if you do like chemistry and stuff. Okay, so now let's move on. Books. Okay, so what are the books that you need to follow in biochemistry? Okay, so Lippincott's Illustrated Review, as I covered in my video on MBBS guidelines of books and syllabus. So books, basically the books that you need to follow. The link to that video has also been given in the description. Basically, all the books that you have, out of those, Lippincott's Illustrated Review, this is the one which I personally found the most useful. Okay, although it doesn't, the only problem with Lippincott is that it doesn't cover the entire syllabus, but it does cover most of the part of the syllabus. So for the part that it does cover, you can of course refer to Lippincott. But again, there's another limitation. Well, I won't call it a limitation, but there's another factor involved in studying from Lippincott, is that it will have extra stuff as well. Okay, for example, you'll need to filter off the extra stuff from the paragraphs that you'll be reading through Lippincott. And how are you going to do that? Remember, I discussed it in one of my videos on I think in embryology videos, I think, oh sorry, in pathology videos I did. explain how in pathology guidelines, basically I explain how to filter off the stuff. You refer to the syllabus guidelines and to the lecture slides. The stuff that is covered over there, you cover it from the book as well. Okay, and the stuff that's not mentioned over there, you may skip it if you don't have time. But if you do have time, you've saved time in other places, then of course you can always read the extra stuff out. Okay, so Lippincott illustrated review, I personally use it. It may not have a few topics as we've already spoken of, so cover them from the lecture slides. Okay, this is one source from where you can cover the extra stuff, or you may find them in Fike, Chatterjee, or Satya Narayan. Now, guys, remember, Fike is actually a book that the Dow teachers do not recommend, but still, it's an excellent book. Okay, it covers almost everything very smoothly. Okay, and it is very concise. And by the way, this is the book that is to be used only and only for the foundation module and for no other module yet during your first year MBBS. Okay, because some facts might be wrong in here, because as our teachers told us that some facts are wrong. So for that reason, you don't have to use it in other parts of the in other modules basically, but still it is a good book for the stuff that it does have the correct stuff. You can just keep it for that stuff. But again, someone might ask that how would we know if the stuff is correct or not? Well, for that, what you need to do is you can cross check against Lippincott or cross check against other stuff. And even if the stuff is wrong, it's just a very tiny bit that's wrong. Okay, or that's outdated, or that has some problem in it, so it won't really cause any issues or any major damage to you. It might cause a minor damage if you are referring to Spike. But again, if you don't want to take the risk, just keep Lippincott and the lecture slides with you. Okay, or keep the Fike. If you are still following Fike, then keep referring to the lecture slides again and again. Okay, but still, I would primarily say that just study from Lippincott, because it has everything covered very nicely. Okay, although it is an extensive book, but meaning somewhat extensive, but again, you can always filter off the stuff. Okay, Chatterjee, on the other hand, is written by an Indian author, so it has a very easy English and it is easily understood. I did buy this book as well, but I didn't actually use it a lot. It is a very extensive book, but of course, you don't have to read everything from it. You just read the stuff that's related to you. And Satya Narayan, by the way, this is a book that was recommended to me by someone, a senior basically, and this is also a very amazing book for biochemistry. Okay, so I would suggest, if you're referring to Lippincott and you don't find some certain stuff on Lippincott, you can check Satya Narayan as well. And by the way, lecture slides are must. Okay, so you have to read through the lecture slides. Okay, lecture slides, you cannot miss out the lecture slides. Now, lecture slides and Lippincott summaries and flowcharts, they are good for review purposes. This is the first place from where you can revise. Okay, after completing your first read of the course, you can give the second read from here, or you can give the second read to the course again, and then a week before the exam or a week before the test, you could just come back to the lecture slides and the summaries and the flowchart given in the Lippincott. Okay, lecture slides must be done thoroughly. And genetics, by the way, this genetics portion, it is very nicely explained in Lippincott. Okay, so for genetics, I would recommend that do not go anywhere else, just refer to Lippincott. Okay, it has the flowcharts and the summaries, all that beautiful stuff in there. It's very amazing. Okay, there's another book called Harper's Illustrations or something like that. Harper's book, this is a very extensive book, and it's a post-graduation level book. Teachers do recommend it to study. Of course, you don't need to study the entire Harper's entire chapters. Of course, the stuff that's related to you, you can study it from Harper. But again, this is a very extensive book, and you might not have enough time because the time given is very limited. So in that case, you may choose to skip this book. Okay, I myself, I didn't refer to it as much as I referred to Lippincott, but I did refer to Harper for a few topics. You can refer to Harper for the stuff that you might not find in Lippincott or other places, or you might find difficult understanding here. Harper might explain it a bit better. But again, guys, you don't actually need Harper over here. You might need it in post-graduation. Of course, you'll need it there, but not here. Okay. So, and even if you are doing Harper, so do it only when you do have enough time left at your hands. Okay. Otherwise, you might end up leaving out some of the syllabus and doing some of the syllabus and end up doing something mixed up. Okay, So Harper's, although it's an amazing book, but it's post-graduation. The problem with it is that it's a very extensive book, okay? And you don't have all of that stuff to do. You've got very amazing, very beautiful books already available to you. For example, the Lippincott book, or Chatterjee, or Fike, or Satya Narayan. Fike, although I'm not Fike actually. Fike is a good book again, but still not recommended by teachers. But still, Satya Narayan and Lippincott, or even the lecture slides are very amazing. Okay, the lecture slides for biochemistry, they're good. They can help you cover all the stuff, and a lot of stuff come from the lecture slides, by the way. Okay, so Harper's, you may refer it only for the stuff that you don't understand. Okay, in other places, so you may refer it uh, in Harper's, but again, do not refer it for the stuff. Do not read the stuff that's not in your syllabus, because again, you might end up wasting a lot of time, because that won't be tested, and you might damage yourself. Otherwise, it's a post-graduation level book, but it's up to you. This is just a recommendation from my side. You may use Harper if you have a lot of time left at your hand. Okay, then topics to be covered. Now let's look at the topics that we need to cover. This is just a brief overview of what we'll be doing, the big things, and then we'll be hopping into the tiny details. Okay, water, there's a chapter of water. It's got a lot of stuff that you've already covered in O level or A level chemistry, actually, or intermediate chemistry. Buffers, you already know the entire concept of buffers over there. Here is going to be a bit more advanced. Okay, the same concept that you've been building since your O levels or A levels or something, or from inter or anywhere. And then in addition to that concept, you'll have some new stuff added on. Then pH, again, the same concept. Again, negative log of H positive ions, the same thing. 4 main biomolecules. Okay, again, you've almost done all of them before entering MBBS. Okay, carbohydrates, proteins, lipids, nucleic acids, the macromolecules basically. Okay, and then enzymes, you've done most of the enzymes. Okay, but the over here you'll do stuff that's going to be a bit more to what you've already done. Okay, for example, the energy profile diagrams have already been covered, but in addition to that, you might find some examples of enzymes, for instance, that are being used in certain reactions, and those examples are important because they do ask examples of enzymes being asked. Okay, and definition of enzyme, of course, lock-in key hypothesis, induced fit, etc., etc. And then coming to gene expression, I've made videos on gene expression, okay, and the link to which I have already given in the description. I have made whole videos on this entire chapter, entire topic, actually. It's a topic, so do check that link out as well. Okay, regulation, gene expression with regulation. Okay, how does the gene work and how is it regulated, etc. Okay, electron transport chain, this is very well explained in Lippincott. Okay, do not go anywhere else for it. Lippincott will have all of the electron transport chain explained over here. An overview of vitamins and minerals, again, you may find it in Lippincott, or you may find vitamins and minerals even in Fike. Okay, even the vitamins and minerals given in Fike is enough. Now let's move on to practical labs. Okay, practical labs, by the way, well, you might find some of your seniors saying that don't take the labs, or skip the labs. It's okay, but guys, I would suggest do take the labs. Okay, do not risk anything, because what if stuff from the labs come in your Viva or from or in somewhere else? Then you'll be stuck if you don't have the information regarding labs. Okay, so they are generally very easy labs, by the way, much easier than the ones which you did in AS or the ones which you did in Inter. Okay, the students, they tend to skip these, so skipping these will not do any good. These labs include serial dilutions. You've already know what serial dilution is if you are coming from an A level background, or you might also know it in Inter. Okay, normal saline, you'll get to know what that is. That's actually the solution that is isotonic to blood concentration. It's a saline 0.9 grams per 100 grams of water of glucose, sorry, of sodium chloride. So stuff like that, you'll just get to it. It's not glucose, okay, it's sodium chloride, because glucose solution is called a dextrose solution. So you'll be looking at making different concentrations of those solutions. Okay, biomolecules detection, proteins, lipids, fats, you've already done those in your previous labs and other easy stuff. Okay, instructions will be given in a manual. Okay, the manual, Dr. Fozia, Professor Fozia of DMC, she has made the manual, and it's a beautiful manual, and you definitely would get it. get one if you get into the DMC, and then you'll be doing the stuff that's over there, and you need to practice them in there. And then you might get labs and internal evaluation. Okay, and by the way, internal evaluation we did not have any. Okay, I myself, batch of D25, we didn't have any internal evaluations. Okay, due to COVID breakdown and then breakout, and then we didn't have any of these. What we had were the modules and why was, etc. Okay, so these are okay, this is Lippincott. Okay, Lippincott's index and the chapters that you need to do, the topics where you'll most probably find your topics of syllabus. Okay, and syllabus is given on the website of Dow, by the way. Okay, now let's just quickly go through these, and then we are done with the video, I think. Okay, so amino acids, structure of proteins, globular. Okay, amino acids and structures of proteins. These are the first two that you need to do. Amino acids. Just a sec. Let me zoom out a bit. Okay, now amino acids and structure of proteins. These are the first two chapters that you'll need to do. Okay, you'll find most of your stuff in these two chapters, and almost the entire chapters need to be done. Okay, the important stuff, you know how to make notes, etc. You'll just make notes and learn them and stuff like that. Chapter 3 and chapter 4 need to be skipped. Okay, these are not in foundation. They'll be covered in blood module. Chapter 5, enzymes, this needs to be done. Okay, chapter 3 and 4, it includes globular and fibrous proteins, and you don't need them right now. Enzymes needs to be covered. Then bioenergetics and oxidative phosphorylation. Remember bioenergetics in your inter or MCAT books? Yeah, it's the same thing, but in a lot more detail. Okay, and this is actually a very important chapter. Okay, and it's pretty easy. Don't worry, it's easy. It's self-explanatory. You could just go and read and you'll get it. Okay, then introduction to carbohydrates, and you might find the same chapter in other books like Fike and Satya Narayan or other books. And then after this, okay, these are the two chapters over here. You don't need glycolysis at this stage, okay? Because again, glycolysis is in a lot detail, which will be covered in other modules, not here. If you want, you may just look at the flow chart of glycolysis, that chart of processes of all those ATP and stuff, but you don't need the details of glycolysis over here, okay? Again, the same goes for Krebs cycle and all of this stuff. You can skip all of this. Yeah, but this chapter over here, chapter 14, this one. In this chapter 14, you've got glycose, aminoglycans, proteoglycans, and glycoproteins. These are three major parts of foundation, okay? Major topics of foundation. You will get most of your biochemistry questions related to these, like a lot of them, if not most, a lot of them, okay? So you need to do this chapter very nicely. And by the way, this is a key point to remember, that you need to skip the synthesis of GAGs, okay? Synthesis of glycose aminoglycans. GAGs is the abbreviation for glycosaminoglycans. Okay, you need to skip the synthesis because that's not included in the syllabus, but you need to do the rest of the chapter very nicely. Okay, after this we'll move on to vitamins over here, chapter 28. Vitamins needs to be done. Okay, again, the names and functions are enough. Okay, you don't need to go into a lot more detail. Okay, and you'll get the idea once you get the lecture slides, or once you get the syllabus, you'll get an idea of what to do. Okay, and then DNA structure and replication and repair, this one, chapter 29, it needs to be done very nicely. Okay, because this is again a major portion of your foundation. Chapter 30, RNA synthesis and processing, this also needs to be done very, very nicely. Then comes protein synthesis, chapter 31, needs to be done very nicely. Then comes regulation of gene expression, and then comes biotechnology and human disease. Oh, you may skip this part, the last one. Okay, you may skip biotechnology and human diseases because that's not included in the syllabus right now. It will be covered later on, but these are the four chapters that you need to do: chapter 29, 30, 31, and 32. Okay, so 29 to 32 needs to be done from here. Then you need to do chapter 14, and actually not 29, it's 28 to 32. 28 vitamins also needs to be done. Chapter 14, and then you've got this chapter 1, chapter 2, chapter 5, 6, 7. Okay, so 1, 2, and 5, 6, 7. So you might have seen a lot of chapters from Lippincott, and still this is not the entire syllabus. You still have some extra stuff like water. You don't see any topic regarding water being covered over here, or regarding other small topics, but you'll get them easily in other books. Okay, for example, now this is Satya Narayan's book. um, in this Satya Narayan's book, if you look at the first chapter, it says biomolecules and cell. Remember that topic that I noted over there, biomolecules and cell. It is covered. Then comes carbohydrates, lipids, proteins, amino acids, nucleic acids, all the and nucleotides, all the macromolecules are covered. Enzymes and vitamins are also covered. Okay, this is a short book, but it has a very comprehensive guideline over here. Okay, a very comprehensive book. And then comes DNA replication, recombination, transcription, translation. You can skip this part. You can do this from Lippincott because it's very well explained over there. But if you want, you must complete it from here. And water and electrolytes is right over here, chapter 21. See, so that chapter can be covered in the bottom is chapter 21. Okay, so the stuff that's missing over there can be covered from here. Okay, additional topics not found in Lippincott shall be done from here or from Chatterjee. You may find the additional topics in Chatterjee as well, water, etc. And from where will you see the additional topics? You'll see them from your guidelines. Okay, and then any book you use, make sure to cross-check the topics from the chapter against the syllabus outline, okay, given on the website of university, any university you are in, okay, regardless of Dow or outside of Dow, but check out the syllabus guidelines. And even in Lippincott, you may find extra stuff. So filter off the extra stuff before reading. And I've shown you, or I've told you the way of filtering off the stuff. How are you going to filter them off? You'll compare against the lecture slides and the syllabus outline. These are the two main points against which you need to compare the stuff given in the books. But still, you may get the stuff from outside the syllabus, but that depends upon the chance. So for that, what students actually do is they read the entire course or entire chapter. Although you don't need to do that, it's a very less likely that you might get something from outside the syllabus. Okay, lectures and lecture slides, especially over here in biochemistry, are very, very important. Okay, so you need to learn the lectures and lecture slides. Basically, it should be done very nicely. Lecture slides almost anywhere are very, very important. Okay, do not tend to miss any. Do give them a read through. Okay, and this pretty much covers up the entire syllabus, maybe. Okay, wait, not the entire syllabus. We still have a few other, like microbiology maybe left. Okay, yeah, this actually covers up all the major portions of foundation module. See you in the next video. And the next video is most probably gonna be on something explaining. Okay, I might be explaining a topic to you, or might be doing some syllabus guideline. And by the way, I do have explanatory videos given in the description, which I made during my first year. Okay, from biochemistry mainly. Okay, thank you guys. Take care.